All right, let's go ahead and fire this up. So let's talk about traditional drug development first. The cost of developing drugs. Okay, now I didn't actually do that, so somehow it moved on its own. Okay. The cost of, is it on automatic?
Yeah.
Oh, okay.
Well, you go and I'll fix.
Yeah. There we go. There's our first technical difficulty. The computer that does the slides is on automatic progression. So let's talk about the cost of developing drugs, first of all. Now, everybody has heard what I think is really the myth of the $2.1 billion drug. Pharmaceutical companies, of course, have an incentive in some ways to record every cost possible when they're trying to figure out how much did it cost them to develop drug X. And the reason for that is that you're probably aware that a lot of people feel that the cost of drugs is too high. And there is frequently a lot of political noise around this, especially during an election year, that the government should step in and somehow freeze that pricing and make sure that the price of these drugs is reasonable. And so the pharma companies, sort of, they estimate their numbers a little bit high, but in reality, developing drugs is very expensive. It's expensive because it's trial and error. In most drugs, when you're looking backward in time, the way that they're developed is either they find some compound in nature, refine it, and then you take this, they're going to test this drug and see if it has a positive effect in your body. But even if it has a positive effect in your body, it may also come with some negative effects. And since it goes all over your body, those negative effects are probably the things that are going to kill that drug. And testing for every possible negative effect is incredibly laborious. It's incredibly time consuming. It's incredibly expensive. And so you get a phase 1 safety test. Let's just put a little bit of this into the patient and see if, out of the 15 healthy patients that we give this to, the safety test is literally on people that don't have the condition that this drug is intended for. So we'll go ahead and we'll put a little bit in, a little bit more, a little bit more, a little bit more, and let's see at what level does this drug make them sick. That's called a safety study, a dose escalation study. At some point they go, okay, it looks like it's safe between here and here, the dosing. Then they move into the phase 2, unless they've had some serious adverse event, because even in a phase 1, somebody who's healthy could die. So, but if everybody, handles the drug okay, some people get sick, they get nauseous, whatever, they kind of know, okay, here's a practical limit for the dosing, and you move into a phase 2 to see whether it works on the condition. So now you're enrolling people that are sick, and you give an acceptable dose to those patients, and you might be dealing with, instead of 15 patients, maybe you'd be dealing with 100 patients now, or maybe even more. And you're going to see, does it actually work? So you put it into these people, and now you're going to do a statistical measurement between the number of people that get a benefit and the number of people that don't get a benefit. And that is your efficacy level. And you hope for something that is significant. In other words, if you've got a drug that alleviates the condition in a significant number of the patients, you're thinking, okay, this thing's on track now. And then finally, you have to go into a phase 3, thousands of patients. And you're going to go ahead and take the dose that you determined in phase 2 that was efficacious, and now you're going to put it into so many people that what you hope is you'll catch anything random that's going to happen. Because what are you testing here? You're testing for genetic background, other drug interactions, diseases that might, react badly to this. So that's what that giant phase 3 is about. And remember, every one of the patients in all these studies is very expensive because you have to monitor them carefully, and you have to do testing on them consistently, and doctor's visits and everything. So that's a hugely expensive process. But it all begins with discovering the molecule, which can also be expensive. You might just decide, okay, we're going to refine things out of trees and test them against, little models that we've developed to see whether something does something positive. Or you might actually, in more modern times, there have been platforms that generate tens of thousands of potential candidates or molecules that might have a therapeutic benefit. And then you spend a lot of time screening that down to one or two that might actually be worth testing in a mouse. And then if it works in a mouse, then you might test it in humans. So even before you get to the human trials, it's expensive. And why is that? Because it's really the random nature of finding the molecule that you want. I call that the Forrest Gump model of drug development. You know, drugs are a box of chocolates. You don't know what you're going to get until you bite into them. So you buy lots and lots of chocolates, and you hope that you're going to find one in there that is good. So that is all changing. But let's talk a little bit about what kind of cost that runs to. So the cost of developing drugs here. You can see that it ranges from $11 billion per drug. This is from AstraZeneca's data, down to about $4 billion a drug. And why is it even more expensive than the $2.1 billion that you hear all the time? That's because they're including all their failures in there. So if you get one drug out of 5 that works, you know, they may say, okay, you have 4 failures at $2 billion each and 1 success at $2 billion. That's $12 billion. So anyway, this is, you know, sort of the bane of that industry. What's the failure rates? This is also critical, right? Because if it cost you, say, $500 million to find a drug candidate, it wouldn't be a big deal if 50% of them made it out to the clinic, right? Because you'd make your money back on that. But that's not the way it is. As a matter of fact, the failure rates seem to be between 5,000 molecules that you might look at and look for. It will eventually result in 1 FDA-approved medicine, right? So what's happening is as you're going through the different phases of your study, things are falling out. They're falling out because people get sick in the phase 1, it isn't effective in the phase 2, or you find some serious adverse event in the phase 3 that's a deal killer. Of course, the further you get through this, the more likely it is that you've got something that's going to make it to the clinic. You notice that in this very narrow part of the curve right here, you know, where you have a very limited number of candidates, it turns out that from phase 3, if anything that makes it into phase 3, somewhere north of 70% of them become drugs. So, you know, the further along, the better. And this also explains how pharma companies like to relate to biopharma companies. So we're a startup biopharma company, and we go out there and we say we have a cure for this or a cure for that, and it's in preclinical or it's in phase 1. And they go, well, preclinical, holy cow, man. Your chances are 1 in 5,000. You know, not even interesting to talk to you. Then we go, hey, we just got into phase 1. And they're like, yeah, only 7% of drugs that make it into phase 1 actually become a drug. That's still too risky for us. Then we say, hey, we got phase 2, we got efficacy. We actually have a result that shows that it's efficacious. And they're like, now it's interesting. All right, now that's called proof of concept, POC. I actually had the former chief strategy officer from Merck told me that POC means proof of concept for a biotech company, but for a pharma company, POC stands for pull out checkbook. So that is the moment when, you know, a biotech company has an asset that they can go ahead and share with a pharma company and co-develop. All right, failure rates is a major killer. What else is a major killer? Development time frames, right? How long does it take to get a drug? This is from an MIT study, average of 12.5 years to get a drug. Now, this is why it's so important to charge as much as possible for that drug, because how much time do you have left if you patented your molecule and it takes you 12.5 years to prove that it works? So how many more years do you have to sell it? 7, 8, 9? Could even be less. So, you know, and what's the rule around any corporation? Profit maximization. So what we need to do is charge the highest price possible for that drug. And what's going to determine the highest price possible? It's going to be whatever the market is willing to pay for it. So the more desperate they are, like if they have a deadly disease or something that's almost intolerable to live with, we can charge a lot, right? Market pricing. That's how everything's priced in the world. So what ends up happening is because there's so few drugs that make it through this whole process, the patient population has a very limited number of choices, and they're choosing between taking what is available or just giving up hope on their condition. And their condition could even be deadly, right? Think about that. Drug companies end up being sort of a group of siloed monopolies. They have these strategic assets like Avastin or Herceptin or Harvoni or Keytruda. And then you've got a patient population that needs it, and you may be the only choice. So what's the price you're going to charge? As much as the people can afford. You're going to try to get every penny you can out of them. And what keeps this non-competitive? Well, the last couple of slides that I showed you, it's basically that anybody that wants to compete with you is going to have to take all that same risk in order to get into the same market as you, right? So 12.5 years of work and, billions of dollars of development cost. So that's a barrier to entry. The barrier to entry is what you look for when you're a company that has a great product, and you might consider something that gives somebody, relief from constant headaches, a great product, right? Then what you want is to be alone in that market as much as possible. So you want to create barriers to entry. All right, you can do that many different ways, but in the pharmaceutical company, the way they do it is the regulatory cost and the cost of developing drugs is so high. That's the number one barrier to entry to competition. Now, what I will posit with you today is that all of that is about to completely change. All right. What if the cost of developing a drug candidate, okay, to get it into phase 1, instead of being $50 to $100 million, so that would be, a reasonably priced drug development in pharma. What if it was $10 million to complete phase 1? That's where AGT is. When we develop drugs, what we've been seeing is we've been accruing information about gene and cell therapies that we can reuse from project to project to project. And as a result, the cost of each project comes down. And we've driven it down to that cost. When we want to do a drug candidate, this may sound unbelievable, that a candidate in a normal pharma development model will take 2 years to arrive at and cost $10 million. We can do it in less than 6 months and less than $50,000. Now, how's that even possible, right? It's possible because of the nature of gene and cell therapy. Gene and cell therapy isn't something where you always reinvent from square one. If you've got a molecule and it doesn't work, what's salvageable about that? There's no great science that explains what it is you need to edit in that molecule to get rid of the adverse side effect, right? So a lot of things end up on the trash heap. But gene and cell therapy, and I'm going to take for granted that most of you know what that is, so I'm going to keep this kind of a light explanation here. But gene and cell therapy is a very, very different process of developing a drug than this random discovery, this Forrest Gump model. Instead, what we're doing is we're designing things from a bunch of components, genetic components that we know work individually, that we can combine and mix and match, and we get a relatively predictable outcome based on what we understand about each one of these components. Now, how do you gain understanding in the components? Experiment, experiment, experiment. And that's why AGT has been around since 2007 and why we're just going into the clinic with our first drug. Because it's better to learn how to program, okay? Because this is what it is, software. It's software because your DNA controls everything in your cells. Your DNA is coded in strings of ACTG, the nucleotides of DNA. The order of those ACTGs determines everything that goes on in your body. You are nothing but trillions of collaborating organic computers that run off of that same operating system, your DNA, and all carry out different functions. Why? How can they carry out different functions? Because the nucleus actually knows what part of the DNA applies to that cell. So that's why the same DNA is in your light-sensitive eye cells and your stomach cells that create acid to digest food, and skin cells that protect your body, and heart cells that, you know, that are muscle or veins or nerves or whatever. It's because all of the instructions that are necessary for every single cell in your body are contained in your DNA, and then each one of the cells in your body executes the part that relates to its job, and then they communicate in this incredibly sophisticated way. Electrically, right? Nerves will pass electric signals around. Chemically, you have cytokines will communicate between cells. Some of them can issue hormones that will communicate with your whole body. It's like one of them getting on the PA system and saying, time to move forward, grow some hair where you don't want it if you're a guy. So, you know, this is the nature of that computer, right? Is that you have a cell run by DNA, the ACTGs, the order of that determines what it does. Why is that? Why do I feel that's like your personal computer that you're watching me on right now? Well, your personal computer just works all in zeros and ones. It's just not as sophisticated as the organic computer. It's base 2 instead of base 4, right? ACTG, base 4, 0, 1, base 2. Also, it only communicates electrically. Wow, I mean, how unsophisticated. As a matter of fact, I bet that your cell phone, your sophisticated cell phone, doesn't even have a command that can replicate itself. My cells have that, so do yours. That's the whole point. This organic computer works in the materials it's made out of. It literally can reproduce itself. That is, you know, call it what you like. Call it Darwin's computer, call it God's computer. The point is it is amazing. And the only problem with this computer is it didn't come with a manual. We literally have to figure out how it works before we can go in there and play with the software. But that's what's happening. We can play with your software drives everything. And this is why we're no longer just doing random discovery. We don't have to generate 10,000 molecules to find something that affects a certain pathway in a certain cell. No, we can design it. That's why it costs us so much less. And the more experience you have at this, the more reliability you have, the more predictability. That's why our costs went down. Our costs went down because we've done it so many times, we were just good at it. We had almost a gut feel for, hey, if that gene product isn't expressing at therapeutic levels, how can we buffer it so it'll express more? How can we flip things around in the viral vector in order to make it express properly? And viral vector, so let's talk for a moment about that. So what's a viral vector? You guys all know what viruses do to your body. They infect the cells. Is there anybody that hasn't heard of viruses recently? So, you know, they attach to your cells, they deliver a genetic construct, it hijacks the cell, executing what? The software that the virus brought. The software that the virus brought is normally a set of genes that just says, make copies of me. And the cell is immediately fooled because it can't tell the difference between messages, these DNA constructs or these messenger RNAs from the virus versus the nucleus, and the virus overwhelms the cell. What happens is, you know, in COVID or in, you know, any type of viral infection, eventually the cell will use everything, all the available materials producing viruses, and it'll fall apart because it'll run out of materials. It's called lysing. So that's what viruses do to you. But what if viruses didn't always bring in something that was bad for you, like instructions on how to build viruses? What if it brought in a gene that you were missing? You were born with a genetic disorder. And what's that genetic disorder? You're just missing a gene and you're not producing a gene product, and the absence of that gene product can be a deadly disease. One broken gene, you could be born with spinal muscular atrophy. You wouldn't live to age 4, and you'd never learn to sit up, roll over, or hold your head straight. You'd die on a ventilator. But if you could bring in the gene that you're missing and put it in all the cells that need it and turn it on where it's needed, you wouldn't have a disease, would you? So if viruses could be repurposed to bring in something good, they could be very useful. And that's exactly what is going on. Viruses can be cracked open. You can scoop out the bad DNA and throw it away. We take HIV. HIV, pretty scary. We crack it open and we scoop out AIDS and throw it away, and we modify it so it can't replicate anymore. And what do we have left over? A shell of a virus that's like an empty stealth bomber. We can put whatever we want in it, something that makes you better. As a matter of fact, later I'm going to describe how we put something in it that cures HIV. But we can go ahead and come up with something simple like just replacing a gene that you're missing, or something a little bit more complex like modifying a cell so it operates better against a disease, or even repurposing cells like little robots in your body. There's so much sophistication to the DNA. We can give cells new features that can be beneficial to you. We are converting viruses, deadly viruses, into helpful updates for the operating system of the organic computer. And that is the software revolution for the next 100 years. It just happens to be pharmaceuticals. And what it's doing is it's driving down the cost, and it looks like either I missed a slide, there it is. And it's driving down the development time, and it's making drug development predictable and reliable. So what we have ended up with is the power to cure, right? Because what am I really talking about here? If we can go in and we can change all the drivers of disease and chop them out, or if we can replace what's missing, or if we can improve the operation of a cell so it's got a therapeutic benefit, well, what can come out of that is incredibly powerful, right? We are tweaking the drivers of your entire body. Now, remember I told you that this computer came without a manual. So we only know so much about how your body operates, but we're learning it faster and faster and faster. And we only know so much about what genes affect what, but we're learning more and more and more about that, right? And then we have limited capabilities for editing your genes. Right now, we have very reliable viruses that will carry new genes into your body, but the new gene could be actually a gene that will install in your body and produce something that shuts off another gene or shuts off multiple genes. So in other words, we have a reliable way to what we call upregulate any gene or downregulate any gene, and there are thousands of diseases that are possible to cure given that. Of course, the monogenic diseases are absolutely just no-brainers, right? If you've got a monogenic loss of function disorder, it just means you've got a broken gene, you're not making something you need. If you can get to the cells that need it, if you can get a virus there, if you can get it to install that gene, you've got a one and done treatment, a cure for life, right? If hemophilia is you're missing factor VIII, well, if we take your blood stem cells and we put that factor in there, all of your red blood cells now will be born with that gene, and they'll make factor VIII, and you'll be normal, right? What if you have lactose intolerance? If we know what it is that you're not producing in your stomach, which we might, I don't know, where would I find that out? I'd just go to the library and look it up. That's the beauty of this industry. If I want to try to find something new to cure, I just go to the library and find out what we know already about diseases. But lactose intolerance, right? We fix that. You're not lactose intolerant anymore. Spinal muscular atrophy is the same thing. Everything from inconvenient to deadly and everything in between that's monogenic, one gene that's not firing, is really just in the wheelhouse of viral vectors. Install another gene. We're actually working on one called phenylketonuria. That's our example in that space. Now, the other thing that's really important about the drugs that we're developing is that gene and cell therapy has the ability to give you a level of targeting that has never been possible before in any other drug modality. Why is that? Certain viruses are what we call tropic to certain types of cells. So you can find viral vectors that are going to tend to go to the CNS, right? Your nervous system. You can find vectors, viral vectors, that'll tend to go to your liver. So what you're doing by having the medicine tend to go one place is that you're getting lower doses everywhere else. And what does that mean? If the place it's going is where you need it, you can ratchet up the power, the therapeutic levels. Because even though it's going to go everywhere else, even though a regular drug would go everywhere else, this one doesn't, right? The virus is going to tend towards one part of your body. The rest of your body's going to get a lower dose or maybe even no dose in some cases. So that is a way to make the drugs more powerful while reducing side effects. But there's another way that's really important. That's called the specific promoter. The specific promoter is really like a smart fuse on the medicine. Imagine if I could put something into every one of your cells that has a cure for something that's needed in only cells that are exhibiting a cancerous state, right? I could hang it from something called a specific promoter, this smart fuse, and I can tune that smart fuse to only turn on in the presence of whatever enzyme or protein I want to look at. And it's only when that smart fuse is triggered that the gene cargo activates. So this gene cargo could be in a bunch of cells that don't need it, but it'll never turn on. Meanwhile, if the specific promoter is triggered, then it will turn on. So how might I use that? So let's say I want this treatment to only go to your liver. I might put an albumin-specific promoter. That's your liver is someplace where there's consistently high albumin and it's low everywhere else. And I might locally deliver it to the liver, so very little gets out of the liver, but anything that does get out, the good news is it probably won't turn on. So then I can do whatever I want in the liver. The other part of specificity is I am working with exact pathways. If I go, the problem in your liver is this pathway, I can sever that pathway or enhance that pathway. That's what the genetic construct does. So specificity is a safety feature, but it also opens up the possibility of changing the treatment between different types of cells in your body to prevent any healthy cells from experiencing the effect of the drug. So let's just talk a little bit about what we're doing with it. We have 3 things in the pipeline. HIV, we've got an HIV cure that we hope to get into the clinic this year. We have phenylketonuria. Phenylketonuria is a horrible disease. If your child is born with phenylketonuria, you find out in that pinprick test where they take a drop of blood from the baby's heel. But if they don't have something called phenylalanine hydroxylase, okay, that's the broken gene in PKU, they can never have animal protein their whole life. And the reason is animal protein has a lot of phenylalanine in it. So does Diet Coke. There's a lot of places that you can get deadly doses of phenylalanine if you've got PKU. But the buildup, since the PAH isn't there to break that down, it builds up and it can damage nerves and organs. And so you have to keep your kid away from all the most fun foods out there, right? They can't eat hamburgers and hot dogs with their friends. They got to be all, non-animal protein to get enough protein. But remember, everything that's done in your body is pretty much done for a reason, right? So the reason you have phenylalanine and you break it down with phenylalanine hydroxylase is because the downstream products are things that are important. And it turns out the important things that come out of that breakdown there, that particular pathway, are a whole bunch of nutrients that your brain needs to form normally. And what happens is these kids are deprived for life for tyrosine and other types of enzymes that go into the brain and make sure that the nerves are healthy, and they become depressed by age 20, schizophrenic by age 30, and suicidal by age 40. As a matter of fact, 1 in 3 phenylketonuria patients will commit suicide by age 40. Think of that, 1 in 3. It's like a slow death sentence from one broken gene. Okay, so PAH, and you only need it in the liver. What could be better, right? We can go ahead and put it in through the portal sinus or irrigate the umbilical vein and put it in that way. And so there's an easy route of delivery to the liver. The liver tends to be like a sponge, so it isn't going to go anywhere else. So we've already got some targeting from that. And then the other thing is we can put specific promoters in there that will make sure that it only turns on in liver cells. And we restore the PAH, you break down the phenylalanine, and you are, for all intents and purposes, normal. So we've got one monogenic disease we're working on. And then here is the earth-shattering thing that I think we'll do in the future. We have a cure for epithelial solid tumors. It is an immuno-oncology approach that I think is really unique, really powerful, really elegant. I haven't seen anything like it anywhere in the world yet. Everybody's heard of Keytruda, and I'll talk a little bit about that later. But this is sort of along those lines where it allows your immune system to help you treat the cancer. But in this particular method that we're using, which I'm going to explain in a little while, we can rely entirely on your immune system. It isn't something we would have to give you in addition to other treatments. No, we can activate your immune system without modifying the immune system itself up to a level where it will clear the epithelial solid tumors that you have in your body. What are those? Breast, prostate, lung, liver, colon, kidney, ovarian, pancreatic, head and neck, and skin cancers, and others. Did you hear any big ones in there? You bet. 900 people a day die from those types of cancers in the United States alone. So 1 in 3 people will have an epithelial solid tumor in their lifetime. Why is anybody's guess? I don't think we have firmly established it. My guess would be, and I think this is reasonable, that what happens is that over time, the accrued stress on your cells, you get more and more malignancy. And over time, your immune system might decline a little bit, and you might hit a critical mass point where your little malignancy doesn't get cleaned up by your immune system. You hear that? Your immune system is working constantly to keep you clear of malignancy, right? And there's a specific type of cell called gamma delta T cells that are 3 to 5% of your circulating T cells, and all they do is try to find small epithelial malignancies and eliminate them before they turn into solid tumors that start growing out of control and could be life-threatening. They're amazing. They're absolutely amazing. They're amazing because 3 to 5% is memory level. These things are active all the time. And the other reason that they're amazing is that even with all of the stresses on you, whether it's physical stress, mental stress, radiation, chemicals, whatever, they will keep 2 out of 3 people clear of epithelial solid tumors long enough for them to die of something else, right? So these things are amazing cells, but once something gets beyond their surveillance, it's growing up, blowing up like a balloon, and it's too much surface area, and these things can't control it anymore. That's when you get surgery, radiation, chemotherapy, whatever. All right, but that's another thing, and it all comes off the same platform. It comes off of everything that we've learned over 12 years of business. And what have we done in 12 years of business? We haven't just become experts in creating this type of software. Yeah, we're artists up in the lab. I mean, they really are. They're scientists, but they're also artists in a way. They've got a sense for what will work. But we've also found commonalities between all these different diseases that we've experimented on over 12 years, and there's been about 20 of them. We've done proofs of principle, okay? In other words, shown a result in a mouse or in a cell model or something like that. And we've been able to figure out, well, what are the common things that are useful in this domain? We pulled them out and patented them, and they now form a tool set, proprietary tool set that we own, that we can reapply and reapply and reapply in order to cure a myriad of diseases. This is something we own. It's something that really took those 12 years to invent, and it's something that nobody else decided to try to invent. Why? Because that's not how drugs are done. Drugs are done by finding something that works, put it in a mouse. If it works, then you run into the clinic and you make $100 million bet. And if it works in the clinic, you're rich. And if it doesn't work in the clinic, you have nothing. That's traditional pharma. But it's really changing right now. You now have software that you can debug. You have software that you can test at the bench. You can anticipate what's going to happen in the clinic. You can reduce the clinical costs. You can reduce other costs like manufacturing, because you don't have to build a pill factory for one drug, because all of our drugs are made in the same type of factory. It's always bioreactors. The bioreactors can be washed out and used for drug after drug after drug. There's a whole industry of people that will rent you bioreactors. So we don't even have to build out manufacturing. All these things bring down the cost of that. And there's a whole bunch of go, no-go points. If you're good at understanding how things should work, you can test them at the bench when it's really cheap, instead of testing them in the clinic where it's really expensive, right? If you get something that's not working in the clinic, you got a lot of work to redo. If you're at the bench and you're sort of at you spend $50,000 and you're testing it in cell models and something's not working, you can tweak it for another, few $1,000s. If it's working there, okay, now you've lowered the risk in the clinic. That's not possible to do with these small molecules to that level or extent. It's possible to do with software because you can logically determine what is causing the thing that you're not happy about or what's underexpressing the thing that you want. And you can chop things out or add things in or move things around, and you can actually improve it and measure it at the bench, and then you can anticipate how it'll work in the clinic. And just to show you the contrast between us and traditional biotech, look at Bluebird versus AGT. Bluebird also has 3 products. They're spending about $100 million a month debugging their products in the clinic. I think they'll be successful. I think Bluebird is a great company. Don't get me wrong. But AGT has 3 products that we're progressing, and it's progressing just as fast as theirs. Now, they don't know that yet, because when we get in the clinic and we get first efficacy and we see the levels of adverse events, I think we're going to prove out that we've leapfrogged their progress on some of their drugs. But we've got 3 drugs going. We've only spent about $40 million at our company total. Sounds ridiculously low, but remember what I'm saying here, because one day I truly believe, and I think this might be in the next 12 to 18 months, you're going to pick up the paper and you're going to find out HIV is cured. And it's going to be by AGT. And remember what I told you, we spent about $15 million on that project. Can you imagine getting to first human efficacy in a drug for $15 million? Part of it is because phase 1 is not just phase 1, it's phase 1 and 2. Why is that? Because the FDA very wisely doesn't want you putting genetic constructs that permanently change a person's body into healthy people. So you don't go out and pay college students to take your drug to see whether it makes them throw up. No, what you do is you prove to the FDA that you have every reason to believe that this is safe and that the risk is worth it because the need is so acute in the population. You can imagine if people have 6 months to live, the FDA's attitude would be, prove it won't cause them pain, then we'll let you try it, right? There's even right to try in some states. But the point is that just makes logical sense. If it's a really hopeless condition and you have a potential cure and you can just show that it won't cause pain, it's worth a test. And you may get a bad effect, you never know, but at least you've lowered the cost to humanity. You're not gonna put it into tons of people and for something minor and find out you get tons of suffering. So anyway, so phase 1 is you put this drug into sick people. You not only see whether your dose is reasonable, but you also get a chance to get some measurement of the efficacy. That's first human efficacy, possibly. We call it efficacy signal that we intend to get in our phase 1, but that's probably good enough that you might see that article in the New York Times or the Wall Street Journal about a cure for HIV. So mark my words, think about how disruptive it is to that industry that spends an average of, depending on the study, between $1 and $12 billion to develop a drug. And they find out, wait a minute, these guys developed something that would normally cost $250 million to get it to that level, maybe $500 million, and they did it in 15 million. I think that that's going to really be kind of a shot across the bow in terms of the old pharma model. It'll be important to engage with companies earlier on that are doing these things, but we're going to establish sort of a new time frame and so forth. All right, so let me talk to you a little bit in specifics about the things that we're doing. And I don't have a ton of time left here, so I'll move a little bit fast. But as I mentioned, we have a experimental cell therapy. So we've developed a cell process that can put your HIV specific T cells into your body at such high quantity and so protected from HIV itself that they can do their normal job of clearing HIV. We can do this even for somebody that's HIV positive. So we can take an immune system that everybody's immune system has HIV T cells. You don't have to be exposed to HIV to have HIV T cells, just like you don't have to be exposed to a cold or the flu to have a few of those T cells in your body that are ready for a cold or a flu, right? It's just that what they'll do is when the cold or flu comes in, they will find it, they'll get excited, they'll kill that viron that they found, and then they'll run back to the lymph nodes and replicate, and now you have more of these T cells, and they start a whole immune cascade that has antibodies, CD8, cytotoxic T cells, so forth, that catches all the variations of that virus. And so, I mean, it's very sophisticated, but it all starts with your sentinel T cell detecting the pathogen, killing the first instance of it, and then starting to replicate and starting this immune cascade. What happens in HIV? HIV has the ability to infect a T cell. So what happens to the first T cell that runs over and finds HIV? It gets infected. Instead of protecting your body, it becomes the beachhead of the infection. As a matter of fact, within 90 days, every single HIV-related T cell in the body is wiped out. Now, we can still get your HIV T cells back, just like we get your cold T cells and your flu T cells back if you have, HIV, because HIV will deplete the T cells, but then when we give you antiretroviral therapy that suppresses the virus, now the virus can't go from cell to cell, and your immune system reemerges from your bone marrow. That's just normal. You may not get as much of an immune system back, but it's sufficient. This is why people that are on antiretroviral therapy, that chemotherapeutic pill every single day, are suppressing the virus enough that they can't get AIDS. As a matter of fact, it's so low, they can't even transmit it to somebody else. So antiretroviral therapy prevents AIDS, and it also prevents transmission. And that's important because in the United States, there's a lot of people not on antiretroviral therapy. Sometimes they just don't want to know that they have HIV, right? They're going to wait until they have a little bit of symptoms of AIDS before they go to the doctor. But they're transmitting it. There's 50,000 new cases of HIV in the United States every year. Think about this. Polio at its peak was only 60,000 cases a year. So this is an epidemic. So it's very important that we get these people to get tested. Now, why don't they want to find out they have HIV? Of course, there's a psychological stigma about it, right? Nobody wants to find out that they're potentially dangerous to everybody around them and that they have this enemy within them that can never be eradicated. I mean, just the psychological impact of finding out, somebody finding out that they're HIV positive is just tremendous, right? Nobody wants to hear that. But it's worse than that, because to suppress that virus and to prevent AIDS and to prevent transmission, you're taking a pill that's toxic. It is going to cause you a lot of the same symptoms, albeit milder, as a cancer chemotherapeutic, because it's a chemotherapeutic. It's a pill, goes all over your body. It does what it's supposed to, and it also makes a little bit of side effects everywhere. So what are the side effects? Nausea, headaches, diarrhea, fatigue. This is common daily side effects from antiretroviral therapy, okay? This is why it needs to be cured so badly. These people aren't feeling good. They may be preventing AIDS, but they're not having the same life of you and I. And so this is a very important thing to fix so that these people feel normal. And, if they were cured, they wouldn't even have the stigma of knowing there was something inside of them that was dangerous to them and potentially the people that they love. So it's important for that reason. But if you're on antiretroviral therapy for long periods of time, then it really gets serious. Early aging, bone density issues, osteoporosis, brittle bones, liver, kidney, heart disease, and extra cancers. So the cost of maintaining an HIV patient in the United States is somewhere between $50,000 and $130,000 a year from the side effects of the chemotherapy plus the chemotherapy itself. Out of that number I just told you, only $20,000 to $30,000 of it is the meds to prevent AIDS. The rest of it are the meds and the doctor's treatment to take care of all the side effects of preventing AIDS. So it's critically important that we fix this problem. So how are we going to fix it? Well, we just restore the natural immune response to HIV. Remember, I told you that the big problem with HIV is that the HIV viron can infect a T cell. So your HIV T cells run to the infection, get infected, and then they start producing virons. They're useless, right? But what if we could prevent that infection? What if we could harden the HIV T cell so that it was impermeable, it was unassailable, unkillable by an HIV viron. That's exactly what we can do. And the way that we do it is we go ahead and make small modifications in the HIV T cell that strips a door handle off the outside of it called CCR5. And it also puts something called siRNA, short interrupting RNAs, against conserved regions of the HIV genome so that we shut down HIV if it even is in the cell already, or if by some chance a version of HIV gets through the cell membrane so it can infect, it won't be able to integrate, it won't be able to produce HIV virons, it won't be able to deplete and destroy the HIV T cell. The HIV T cell will go on and keep doing its job. So this is critically important. So those are the construction people. I don't know if you can hear it. They're out there vacuuming. So the point is that if you can correct the initial problem in HIV, if you could take away the advantage of HIV against the T cells, against your immune system, the immune system is capable of clearing HIV just as easily as any other virus, colds, flu, whatever. That's the point. So how do we do it? First thing we do is we draw out what's called a leukopak. So we do a blood draw on a patient and we send it through a filter. So what it does is it gathers all the T cells, the white blood cells in that blood. And we have to take about 400 milliliters of blood that's highly concentrated for T cells. So what are we getting? That's called bulk T cells. We're getting every type of T cell. There's some HIV in there in very, very low amounts. But then there's cold, flu, bacteria, fungus, cancer, every type of T cell that you can imagine. So the next thing that we're going to do is we put it in an automated machine. Okay, because what we're trying to do is reduce the cost of delivering this therapy so that it's practical to get to as many people as possible. If the cure costs $10 million, how many people can afford to be cured by HIV, of HIV? Very few. If the cure is $500,000, hey, that's about the price of treating them for 5 years. Now we're talking practical, right? Okay, so we have a machine that does all the work for us, and what does it do? First thing it does is it dumps in some what are called peptides that will stimulate the GAG-specific T cells, which are HIV related. That proliferates them in the mix, and it activates them in a way where then we can very easily deplete out all the non-HIV T cells. So we have something that ends up being a mix of 35% to 65% just HIV T cells. Then we go ahead and make our modification. Why do it here instead of back here? Less T cells, right? Now we're only modifying HIV T cells. This has been a problem in a lot of therapies. Vector costs a lot of money. If you look at the last attempt to do something like this, they were using $500,000 worth of vector for one patient, and only 1 in 10 patients was getting well. So that's a lot of money for one cured patient. Here, we're actually using $35,000 worth of vector. So you can imagine how much cheaper we can do this whole process. There's other costs in it, right? But the total cost of this is roughly about $100,000 today at retail pricing. So you could sell it at $500,000 and make a lot of money, right? So if a pharma company got this product, they'd be saying, yeah, this is right in our sweet spot, 80% gross margin, right? All right, the other nice thing about the machine, anybody can buy one of these machines. It's $250,000, doesn't need a clean room. It's all automated, push a button, and it runs. What comes off the end of this? So after we modified the T cells, then it goes into a culture, still in the machine, and it amplifies those cells up to lots and lots and lots. We want some number where the T cells have a major advantage over the disease. And what's that number? Well, you could ask only, any immunologist, like what number of CD4 positive T cells do you have after you clear a virus successfully? And guess what they'll tell you? Roughly 100 million. This is a number that's sort of known in immunology. So what do we end up making? 1 billion, 10 times that many. Now we know that's a safe dose because guess what? If you've heard of CAR Ts, there's lots of experimentation going on in CAR Ts. It's another type of process like this where you pull out T cells and modify them. It's a more toxic modification. It's done on a larger number of cells, and then they reinfuse those cells. So we know that you can reinfuse that volume of cells safely. So we have 1 billion of these cells, and then the last step is exactly that. Go ahead and reinfuse them into the patient. They should at that point be able to throw away their toxic chemotherapy, their daily pill, throw it away. Why? Because as the virus reemerges from their latently infected cells, from the viral reservoir, guess what's there? 1 billion HIV T cells ready to react against it that cannot be infected by or depleted by HIV. They just do their job. They just kill that stuff. Now, I have an incredible confidence level that this is going to work. And why is that? Well, it's because we had to, in order to perfect that automated cell protocol, we had to actually put we had to find 12 different donors to give us blood, HIV-infected individuals, run it through that process and get the blood product. Now, we can't put it back into them until the IND is approved, until the FDA says it's okay. And so what we end up doing is testing it, what's called in vitro or ex vivo. We can put it in a petri dish and let's see how much HIV it can clear. Well, the amount of HIV that it could clear was orders of magnitude better than anything ever seen, including antiretroviral therapy. So we know it's got the clearance power that it needs. And when you run the numbers, it's got enough to clear the entire human body. As a matter of fact, we think it's 3 to 10 times more potent than it needs to be. But don't believe us, because we showed that data to NIAID, National Institutes of Allergy and Infectious Disease. Anybody heard of Tony Fauci recently? This is his department, his right-hand man, repeated our experiment. They bled their own patients, they got their own blood product, they challenged it with HIV themselves, and they validated what we saw. As a matter of fact, their results were a little bit better because they have so much more resources than we have. So this looks really good. And they actually co-authored an article with us that appeared in Molecular Therapy. And it explains this whole process, and it shows you, I think, the future of HIV. But this shows that this has significant impact against HIV and is likely to lead to what? A functional cure. In other words, functional just means as good as. You may still have a little HIV in your body for some time, but you can't progress to AIDS. You can't infect anybody. And here's the best news. You can never be reinfected. Can you imagine these people going from being HIV positive to knowing that they not only got rid of it, but they can't be infected again? Be dream come true. Let's talk about immuno-oncology, and I mean super fast. I literally have 4 minutes. I will stay for whatever time you want to answer questions, and I apologize that I am so talkative today. So you get a tumor, right? What does this mean? It escaped your gamma delta T cells, right? Your gamma delta T cells eat all the little malignancies, but if you get unlucky and enough of it grows into a tumor, it's too big, the gamma delta T cells can't keep up. But what we do is we just put a tiny little bit of a proprietary lentiviral vector that carries a transgene that overexpresses a stimulatory small molecule that calls in every gamma delta T cell from your entire body and activates them. When they're activated, they eat at 300 to 600 times their normal rate. Now you notice here, we've only modified the cancer. We didn't modify your immune system. So it's very, very safe. And as a matter of fact, gamma delta T cells, the ones we're stimulating, there are diseases where these T cells can become 95% of your circulating T cells and you don't die. So these things are safe at very high levels, and they've never been seen to eat anything but epithelial malignancy. They are perfectly targeted with an NK receptor. So they all zoom over to the tumor. You see those red cells in there? When we put the vector in, it's not going to get all the cells. We don't have to. All we have to do is get about 20% of the cells. That starts this catalytic event where all the T cells rush in. That's shown up here. And then they react to the stimulatory small molecule and they become activated. What happens when they activate? They just go ahead and turn right on that tumor. And what happens to it? It's obliterated. Makes sense. But here's the side effect, and it's good. What else does it find? It finds the secondary tumors and metastases, because these things are circulating all over your body in that activated state. Everything smaller than the primary tumor will also get eaten in the time it takes to clear the primary tumor. Primary tumor is almost like a timer, right? Because once the primary tumor is gone, the stimulation is gone and you're normal again, minus the tumor and minus all the secondary tumors and metastases. And as a matter of fact, what we're finding out is you could come in for a lung tumor and we treat you for that. If you had a little bit of breast cancer or a little prostate cancer they hadn't detected, it'll mop that up at the same time, because the gamma delta T cells don't know the difference between breast, prostate, lung, liver, colon, kidney, ovarian, pancreatic. They are the cells that keep you clear of all of that. Our collaborator on this is the Stanford Medical School. We think we'll be doing a clinical trial at Stanford with our collaborator, Dr. Dean Felsher, in late 2021 or early 2022. We have 1,000 mice in late stage cancers, human cancers, and we take human gamma delta T cells and put them in their body. They do nothing. We go ahead and inject our viral vector into one of the tumors and all the tumors disappear. 85% of the mice come back completely cancer-free. This is really promising preclinical data, and it translates beautifully to humans because it's all human. Everything, the cancer, the gamma delta T cells, everything's human. So, what I want to tell you, and this will conclude my presentation, high tech is coming to biotech. Developing drugs is more like developing software, right? We're not trying to discover or do things out in nature and hopefully find something that's useful. No, we can now design things that go into your body and modify them in very specific, powerful ways to cure a disease. And the costs are dropping like a rock. This is true in every single aspect of gene and cell therapy. This thing here is just sequencing of the human genome. Remember Moore's law from computing? Power doubles every year and cost halves. Look what sequencing did compared to that. It was pretty much on track for a while, and then it just fell off like a rock. So this is actually happening faster than traditional tech. I've been part of many tech revolutions. I'm telling you, don't underestimate what gene and cell therapy is going to do to disrupt the entire pharmaceutical industry in a great way for patients, in a great way for society. Could be a great way for pharmaceutical companies if they can figure out how to embrace this stuff. A lot of them are late to the party. A lot of them are trying to work this thing into their traditional model. They're trying to figure out, when do we engage with an asset? They're going to be coming in too late. And the funny thing is some of them are overpaying for stuff because the problem with software, highly competitive. You can't count on 19 years on a patent. There's more than one way to do the same thing in software, right? Just because somebody wrote VisiCalc didn't mean that they owned the spreadsheet market for 19 years. No, they got wiped out by Lotus 123. They also didn't own it but for 19 years. They got wiped out by Microsoft Excel. That's the point, is that innovation and competition are going to be part of this industry. It's no longer going to be about holding a patient population hostage and getting as much money as you can from them. You're going to have to give them value. In other words, people are going to buy drugs based on what's giving you the most value per dollar, the most life per cost. That's what people want. You're not angry at Samsung or Apple for the iPhone, are you? It's expensive, but it's worth what you pay for it. The value you get is more than what it costs you. That's why you're not writing your congressman saying they should be putting price controls on cell phones. There's lots of alternatives. There's lots of competition. Even Excel. You practically can't live without Excel, right? But if they charge you $10,000, you'd learn to live without it. You'd use Google Docs or Linux, right? That's the whole point. This is totally different, all right? And pharma companies are going to have to figure out, how do they move into this new world when now 10,000 different diseases are accessible in gene and cell therapy, and they are not just treatable anymore, they are curable. There's a form of blindness cured by Luxturna. There's spinal muscular atrophy, that terrible one where you die by age 4, cured by Avexis, now owned by Novartis. 2 incurable cancers cured. XSKID, ACA SKID, which is bubble boy disease. It's you're born without an immune system. Curable, right? You can't do that with a small molecule. You can't do that with a daily treatment. And these are one and done cures. You put the gene in and you change them for life. Fantastic stuff. It's a very hopeful, wonderful time in biotech and in pharmaceuticals and in healthcare in general. And, you know, we're very excited. I think we're a leading company in that. But there's going to be a lot of companies going into this. You're already seeing it. Gene and cell therapy is hot from an investment standpoint. But this is why, you know, we invited you to this seminar. You know, the idea is how do you balance out your portfolio now? Well, at least now you can see the future that's coming. You can think about it. Next year, when you see some results from HIV, remember what I told you here, because things are going to change fast. All right. Thank you so much for attending this webinar. Really, it was wonderful to see all of you. Wonderful that you came here. And like I said, I'll just stick around and I will answer questions for a little while, or a while actually. I love to talk, so no problem. And but I understand people are going to drop off. Again, thank you for coming. And hopefully we'll see you again. There's lots more to talk about. I only got to about half my presentation, believe it or not. I got many more things to tell you that I'm sure you would find fascinating. We'll invite you again. Thanks. All right, we got any questions? Okay. Oh my gosh, that is a long question. I think we need a shorter one.
Everyone is able to unmute themselves at this point.
Yeah, you can, everybody can unmute themselves. I'll tell you what, go ahead and somebody, you know, speak up and I'll try to answer your question. Go for it.
I have to drop off. I just want to say this is Richard Alvarez from Brain Institute. That was fascinating. Thank you for doing this.
Sure.
I apologize. It's a sign of the times. My daughter just.
Oh, that's...
But thank you very much. And it was wonderful listening. Thank you.
Thank you very much for coming and for your encouragement.
Absolutely.
Appreciate it.
Thank you.
Anybody else? Compliments are welcome. Yeah, thank you. I mean, if that's all you got for me, I'll take it.
Hey, Jeff, Gene Valentino here.
Hey, Gene.
Hey,
how you doing?
For those guests I don't know, I'm invested in AGT, and so I've listened to Jeff talk maybe a dozen times now, and every single time he does, I'm learning something new and different. One of the things that stood out today that I thought was fascinating was the issue of your INDs are underway and you're waiting for clinical trials to begin. But with this story you've just described and the compelling evidence of success that's pending, it would seem that they'd be rather quick, especially in the world scene with all these other virus issues going on. It would seem that they'd be willing to step up and give you that approval for clinical trials sooner than later.
Well, so the problem with the FDA right now, you know, sort of the difficulty in working with them is that because gene and cell therapy is proving to be so powerful and, you know, such a revolution, there's so many people jumping into it. It's like a .com boom. I call it the .gene boom, named after you, Gene Valentino, right? So, but the point being that tons of money is flooding in. And even though some of these people aren't taking the time to really vet their ideas, they're still submitting INDs. It's like a gold rush, right? So the FDA is overwhelmed. And there's probably like a handful of people that are qualified to actually read a cutting edge IND in gene and cell therapy, right? And if they want to read ours, they have to also be experts at HIV. Where do you find these guys? If you can find them, send them to us because we'll hire them. That's the whole point, right? So they are overwhelmed. So they got in this mode where they had to put you in a holding queue, but it's illegal to put you in a holding queue. So they have to ask you questions every 30 days and put you on hold again. So, you know, we've been getting random questions for 7 months, but now we finally seem to have made it to this really sensible review cycle, and everything that we're hearing from the FDA now makes sense. They're reasonable questions. They seem just as passionate about getting this into the clinic as we are, limited by safety. They're not saying anything that sounds crazy to us. They're not asking us anything where we're like, why do we have to teach these guys virology, right? You know, why do we have to give them directions on how to find the bathroom down at the FDA? Can't they find that on their own, right? I mean, this is the kind of questions that were showing up in the early reviews. And, you know, so it's just, it's a night and day now. So I think that we got we put a submission in about, when was that? July 10th. So they have to answer us by August 9th. But in the meantime, they sent us some clarifying questions that also made sense. I mean, they really seem interested in, you know, trying to get this thing done. That's great. This is what the FDA, look, and look, I got no hard feelings towards the FDA. You know, the thing is this, they're responsible to make sure that you don't accidentally get snake oil or something that kills you.
Yeah,
right?
but it's
They
fair to
really
say,
seriously.
it's fair to say that these other competitors and everyone rushing to the gold rush here, if you will, are certainly not enjoying the entry cost that you seem to have managed pretty well.
Yeah.
I mean, you mentioned $500,000 to get to clinical trials, and you're a tenth of that, I presume, or something.
Yeah. No, we're a fraction of the cost that they're enduring. And our overhead is really low because we really stick to our knitting. We make genetic constructs. We don't have manufacturing. We just go out and get a CMO to do that, right? So, you know, we can wait them out. This is my most proud moment. If they approve the IND, we will have outlasted the FDA. We outweighted them. I think that's pretty exciting. So anyway, yeah, that's the situation. I think that's looking good. And like I said, my confidence level that we're going to cure HIV is 99%. Okay. So take that as an entrepreneur's 99%. So it's probably more like 90% in reality. But, you know, there's every reason to believe that this is going to work. And why? Because there's been experiments where they've gotten cured patients on similar theories, but the reliability and the cost made it completely impractical. The cost was $500,000 in vector per patient, and the reliability was 10%. Now we're delivering 2,000 times the potency of the best case of that study that I'm talking about. 2,000 times at a fifth of the cost total. I mean, the vector's like less than a tenth of the cost. So, you know, this is tremendous difference. And I think that from what we're seeing in the blood products, we should get a high percent of the patients getting durably in remission. That's what you look for. It's called a functional cure. They are able to throw away their meds and their virus doesn't rebound. And you find that out pretty quickly because if somebody stops their meds, who's on antiretroviral therapy, they'll rebound to full viremia in 21 days. The whole infection cycle will restart. All of their HIV T cells will run to the pathogen, get destroyed, and it'll take over their body again. So, any other questions?
Jeff, I don't have a question, but I do have a comment.
Mm-hmm.
This is Lance Webb. Last time we met was when we dedicated Roscoe Brady's portrait in your boardroom. And
right.
I
How
just
are
want
you?
to tell, very good, Jeff. And it's a fascinating presentation. And I think Roscoe, knowing him as well as I did for 25 years, he would be extremely proud of the work that you're doing. You know, it took him 30 years and a lot of failures at the NIH to bring forth the first ERT for rare genetic disease. And it looks as though you've almost found a free way to bring therapy for unmet needs. So I'm proud of what you're doing. I know Roscoe would be, and you and the whole organization are a great bunch of people. So just keep up the great work, and I wish you the great success.
Thank you so much. Yeah, just to, you know, so everybody understands why Lance is bringing up Roscoe Brady. So the way I got turned on to gene and cell therapy is that I came out to look at a business plan from somebody in his lab at NIH. And that person introduced me to Roscoe Brady, who told me about his cutting edge research in viral vectors. And, you know, even though I'm not a biotech person, I could recognize from all of my tech experience that what he was telling me was that we can now take deadly viruses, crack them open, and convert them into updates. Viruses could now make you better. And that he was along that path of making that practical. He was actually doing some of the first research in lentiviral vectors. I mean, when I saw that, my head practically exploded. I remembered enough high school biology and how DNA impacts, your health, that I told him right there, I'm like, we may be able to send radiation and chemotherapy the way of bloodletting and leeches where it belongs. It was a really exciting day to meet Roscoe. Such a great guy, so I didn't realize what an accomplished, globally recognized, decorated scientist he was, because he was so down to earth and so kind to me, and just so passionate about curing things that he just took the meeting, right? He was willing to meet with anybody that shared his interests. And so that was an inspiration that started the company. And he was retiring. At that point, he was in his mid-70s. And NIH said, yeah, if you'll continue his research, you can just have everything in his lab. Hire the postdocs if you want. That started the company, free. I got the most cutting edge research in viral vectors and the best plasmid system on the planet for free. And so I had some money. I was retired at the time. I had been successful in Silicon Valley in computers, software, IT, internet, right? I was a tech guy. And so I just thought, this is too big. I had this vision for a revolution in pharmaceuticals. This was back in 2007. I met him in 2006, I think. 2007, I was like, how can I not get involved in this? So I started the company. And my dad, who's on the call here too, you see Aaron's iPad, that's my dad. He and I put tons of money into this thing to keep it alive for 8 years until anybody else cared. And then thank goodness we had really great, high net worth individuals who, I could talk to about my vision for the future. And they were like, that makes sense. We'll risk a little money on that. And it's been a snowball rolling downhill since then. We've been just hitting more and more milestones. So Roscoe Brady unfortunately passed away in 2016. So he was he was our lead scientific advisor, chief scientific advisor, until he passed away in 2016. And Lance was kind enough. He works for Genzyme. Now, why Genzyme? Roscoe Brady had invented a cure for Gaucher's disease that NIH gave to Genzyme, and Genzyme ended up selling $1 billion a year of something that Roscoe had pioneered, enzyme replacement therapy, saving lives. This Gaucher's disease was one missing gene, which meant one missing gene product, glucocerebrosibase. This is what Roscoe discovered. And then Roscoe figured out how to make it in bioreactors, and they could put it into the patient. And if they put it into the patient, they didn't get the deadly consequences. And what were the deadly consequences? If you didn't have glucocerebrosibase, one gene product, your organs would absorb lipids, swell up, and burst. Everybody died by their teens or 20s. And thanks to Genzyme, thanks to Roscoe Brady, those people lived normal lives. It was painful to take a shot every day, but that's why he started looking at viral vectors. He said, is there a better way to fix one broken gene than to put in a shot in the person's body every single day for the rest of their life? And that's when he thought, maybe these viruses could carry a one-time fix for that gene. And that's why he was going down that path. That's when I met him. It was late in his career. He was going to shut down his lab. I was just, a really fortuitous meeting, but coming from my tech background and seeing this new technology and having this immediate vision for the future, which now 12 years later, I think almost everybody sees that future. They may see it differently than me because I've been doing it for 12 years. So I know it in more nuanced, more detailed way. But the reality is I think everybody accepts that gene and cell therapy now is providing cures for the formerly incurable, treatment for the formerly untreatable. And, that the efficacy is high, the safety is high, the adverse effects are low. This is going to be huge from a technical standpoint. I think one of the things that I bring to the mix is that this is huge from an economic standpoint as well. It makes sense from a value perspective that these things, they sound expensive. $500,000 to be cured of HIV until you realize that you're spending $100,000 a year to not be cured by HIV and feeling like crap the whole time. Now you go, yeah, 5 years, Gilead could buy that drug and they could sell it for, $100,000 a year for 5 years. The insurance companies wouldn't even have to change their normal payments. The only difference would be 5 years later, that HIV patient wouldn't cost them anything anymore. They'd save 20 years at $100,000 a year. So it's high value, even though it sounds expensive. Yeah, $20,000 a year sounds cheap until you realize it's $100,000 to treat this patient from all the comorbidities of the antiretroviral therapy. That's even ignoring just the quality of life issues, which I think are really, really important. I mean, this is what AGT was all about. It was founded to leverage the power of gene and cell therapy to reduce suffering, reduce early death from serious human diseases. And what I wanted to do was to implement something that would be a whole new level of efficiency in drug development so we could bring this out to the widest audience possible. And I think we're doing that now. HIV will be the proof, not just of that HIV can be cured, but that the model works and that our costs will be evidence that these diseases can be done and cured at practical prices, even to serve small audiences. What if I could cure a form of blindness and it cost me $20 million to develop the drug, but only 1,000 people had that disease? That would be a really small disease, right? If there's 1,000 people times $100,000 is what? Somebody help me out with this. So it's a lot, 100 million. That's a profitable drug. I made $80 million, right? You know, I mean, and remember, I only have to produce it. And where do I produce it? I can buy the materials from a CMO. I don't even have to have my own manufacturing, right? Small, you know, monogenic diseases are going to get cured at faster and faster paces going forward. Because I think that, you know, there are stumbling blocks to each one of them. It's not absolutely trivial. Everything has a challenge. PKU had its challenges. We've knocked those things out at this point. There's a reason why Bluebird is having such a hard time getting their hemophilia and their beta thalassemia drug out, right? You have an immune response to the new protein. That's a tricky problem, right? But in phenylketonuria, we figured out a way around that already. So, you know, there'll be challenges with each one of these diseases, but we'll discover little keys to unlocking whole new swaths of cures for different types of diseases. That'll become part of our tool set. Our tool set will eventually become like an iPhone, right? That does all the heavy lifting. And when you want to cure a disease, you just write an app on top of it. And you can rely on all of the things that deliver the goods into the right place to cure the disease. We'll drive the cost down. And by the way, we'll license it to the world. You know, there's not a reason. We can't do every single disease. There's 10,000 diseases that ultimately will be cured by gene and cell therapy. And I mean cured, not treated. It's really, really different. So we can't do them all. You know, we're going to do HIV. We've got infectious disease, a bunch of things going on in that. We're going to, you know, possibly be able to be successful at this immuno-oncology thing. That's huge. That's $1 trillion market. That'll all keep us busy. But what I'm hoping is that collaborators will come to us, you know, from biotech and maybe even from pharma, and they'll pick diseases where they know exactly what pathway needs to be modified. They can talk to us and we can tell them, yeah, whether we have the tools for it or not today. What's the beauty of this industry? It's just like computers. If you don't have the power or the tools that you need today, just wait. Somebody will find it, right? So you got doubling of your capabilities every year and halving of your cost. And what's possible in that situation? Anything. If you can dream it, you can do it. If you wait long enough, the solution will appear. It'll be some little element you didn't have before where you go, oh, I can combine that with this and this and this, and I can cure this thing over here. So, you know, the first computers couldn't drive your car or fly your plane. They're not having any problem with it now, are they? Now they talk to us and they sit in our pockets. That is the nature of tech. Tech has come to biotech. High tech has come to biotech. This is a whole different way of developing drugs, completely different economics. Everything's different. All right. If I don't get another question, I'll wrap it up for the day.
You guys really.
Jeff, I just want to say,
Oh,
Jeff, I just want to say thanks. It's Kirk Hallbeck out here on the West Coast.
Hey, Kirk.
Great presentation. Hey,
Thank
how
you.
you doing?
Thank you.
I would love to get this out to this presentation out to some of my friends and family as soon as I can. So have Norman get cracking on that for me, because
Oh yeah, yeah. I think it's. I see up in the corner. It looks like it's recording. So yeah, we'll put it up somewhere and you can get to it. Thank you.
okay.
All
Thanks
right.
again, Jeff.
You're welcome. Thanks
All
for
right,
coming.
bye-bye.
Jeff, enjoyed your presentation. Can you tell me, are these genes hereditary genes, and is there going to be issues with religious and regulatory organizations with that?
Well, you know, I'm not a theologist or an ethicist really, but what I would say is that, yeah, this is pretty powerful stuff. You know, ultimately, today it wouldn't be practical, but, you know, 50 years from now, 100 years from now, designer babies, making new animals, you know, it's all going to be possible. But, you know, that's the way it is with all technologies. There are always two-edged swords, and sometimes they bring up policy decisions and even theological things, right? You know, it's nuclear power could light your city and keep you warm, or it could, you know, evaporate your city and you know, and kill millions. An accident can cause a problem. We had accidents in the early days of viral vectors and a few people died. You know, that threw a pall over the whole industry. But what I'd say is that, you know, all this stuff is a personal decision, because there's a difference between faith and the law. The law allows this stuff so long as you're not hurting people. That's the whole idea, is we can coexist in the United States under the rule of law. People can make their own decisions. We're not going to force anybody to take a gene therapy if they're dying. If they think that's not the right thing to do, they don't have to take it, right? But if somebody's dying and the gene therapy, you know, could knock back their liver cancer for 10 years and they could spend 10 more years with their family or going back to work, you know, having a life. If we could go ahead and use gene therapy, you know, to actually correct things that are just wrong in your body and end suffering. Personally, I think it's ethical to do. And then it's going to have everybody's personal decision. Yeah.
Right. Well, enjoyed your presentation. Thank you so much.
Thank you. Thanks for attending. I really appreciate you tuning in. Thanks.
Jeff, can you hear me?
I can. Is that Arun?
Arun, yeah.
Arun, yeah.
My
Hi.
name's Arun. Hi. I just want to say that his presentation was really inspiring. I recently was diagnosed with HIV this year, and it's been overwhelming hopelessness. And you've really given me a lot of hope, and I believe in you. I believe in your company, and I'm looking forward to the day where I read about it, how you guys have done it, cured HIV.
Oh, thank you. Thank you so much. And I think it is a hopeful future. I think that, you know, we got a very, very powerful new technology. It's going to take a little while to get to everything.
Yeah.
But it's really like a collision between three giant planets right now. And AGT is trying to live in the middle of that. It's understanding the mechanism of action of disease, understanding the genetic underpinnings of that, and then having editing tools to correct it. And many, many things are going to happen at that collision point. Right. And it's coming on fast. So yeah, I wish you the best. And I hope that we'll be able to, you know, either do something related to you ourselves, or that somebody will come to us one day thinking, hey, that's a great one to go after. And our tools will provide a practical solution where the first day that they start experimenting, they're 80% done with the drug, and they could enjoy a $10 million path to the clinic, you know, like we do.
Yeah. As a Canadian, is it possible for me to be part of the cure the trials, the clinical trials, or it's not? It's only for U.S. citizens.
Well, so clinical trials is a whole separate thing, because we don't have any control over the patients. You have to enroll with the hospitals. And I don't think that you have to be a U.S. citizen, but you have to be a patient of a doctor that's at one of these hospitals that's enrolling. And I don't know the details of it, but if you shoot us an email, we can give you details.
Will do.
And I'm sorry, because you were so quiet, I didn't hear what your disease was. I thought it was some monogenic disease, you know.
No, HIV.
Oh, you do? Oh, okay.
Yeah,
well,
yeah.
Look, it's coming. It's going to come to a clinic near you anyway.
Yeah.
You know, don't panic about it. Just stay on your ART. Keep your HIV T cells growing and healthy. And, you know, here's what my hope is. is that, you know, step one, we show that we can do it in the United States for insured patients. Step two, we drive the cost down by 50% over 2 years. Step three, we drive the cost down by a factor of 10 over 5 years. All right. And because this is a machine, it's done in an automated cell process that, you know, is, anybody can afford that machine, $250,000. Any hospital or clinic can get it and start curing HIV patients. So it could go anywhere in the world. All we need to do is send our proprietary viral vector, you know, as doses for the machine. And then they just leukapheresis, press a button on the machine, you know, do the release criteria on the cells to make sure they're healthy, and then reinfuse them into the patient. And, you know, I can't guarantee it's going to work in this first round, but my confidence level is high that we have something that is likely to lead to a functional cure. And also remember, cool thing about software, it's debuggable. There's been lots of examples in gene and cell therapy where the first one didn't work and you tune some things up and the second one did work. So the percentage of these things that are going to make it out there are going to be high. We've tried to do as much testing as we can in cell models to assure that this is safe and effective. And so that's why I'm so confident. And then NIAID got the same results that we did. That also boosts my confidence. So coming soon to a clinic near you. All right.
Plus, it's the way the Berlin patient and the London patient were cured. So, you know, it's looking good. And all that sounds great, man. You know, thanks a lot. Thank you. So.
All right. Yeah, hang in there. And thank you for tuning in. I appreciate it.
Hi, Jeff. This is Sanjay Srinivasan. Great presentation, really excellent. And I completely agree with you. I'm based here in Richmond, Virginia, and I work with several early stage startups. One of the companies I'm working with, we are also into immuno-oncology using cell therapy, using viruses, and working with, working to treat lymphoma and lung cancer and various tumors. We are basically looking at doing the cell therapy for 1/10 the cost of CAR T, with essentially no side effects, like dementia and cytokine storms. And this can be done at any outpatient center rather than going through a very rigorous, cancer treatment center. So again, we're also happy to chat with you, Jeff, and get your perspectives and thoughts. But
sure. Yeah, it'd be great to hook up.
Yeah,
it's fun to talk about this stuff. I mean, people that are in the industry are very excited, and with good reason. And it's always nice to find another person who is involved in this and excited by it like I am.
Very nice.
So thank you for coming today. Appreciate it.
Thank you. And best
All
of
right.
luck.
Well, I see, it's going down gradually. We still have 30 people in here. So I see Glenn Hartstock raised his hand. Did you have a question you wanted to ask? I don't know if your hand is raised and you're not muted. So, but all right, you may not have meant to do that. Anybody else have any questions before we call it a day? All right. Look, thank you so much for coming, and for all these people for staying late. I really appreciate that too. It was really fun speaking to you, and I hope I'll see you again soon. All right. Take care. Have a great day.